Thanks very much for the introduction and also for the invitation. It's such a huge honor for me to stand here and update our work. The first part, actually, two parts from my talk. First part is update of my Harrington project and what we have done, what we have delivered in the last 3 years. And second part will introduce everyone UPNAT, UK platform of nuclear acid therapy for rare disease. Fingers crossed. Yes, it works. The disease is SPT-LC1-related sensory neuropathy, and it's a severe peripheral neuropathy, mainly affects the nerve ends. And it's a rare disease, and the gene is, related gene is SPT-LC1. So here, we focus on SPT-LC1 and also S2 also involved as well. So in the UK, we have a group of patients and affected by a particular mutation. It's a UK founder mutation. It's a C133W. So over 70 adults and over 200 children that are affected by this mutation. And all the group of patients are under the care of Professor Mary Riley at Queen Square. So, and then this is geographically, this mutation also been spread to America and Australia, some other countries as well. So globally, about 200 adults affected. So focusing on that disease, because there is a very urgent unmet need for new treatment. And this is a dominant gain-of-function mutation. And the mutation in SPT, this is serine palmitoyltransferase enzyme. This enzyme metabolizes serine and produces sphingolipid, which is very important for neuronal function. A mutation in SPT changes the substrate of the enzyme from serine to alanine and produce toxic product metabolites called deoxysphingolipid. Because this is completely new to our body and there's no enzyme existing to get to further metabolize, get it, clean it out of our body, and the accumulation or increased level of deoxysphingolipid, short for DSP, is neurotoxic, and that's how it reflects in clinic with this neuropathy. So together with Professor Francesco Montoni, we come up with the strategy. It's called allele-specific antisense approach, and to particularly target the mutant transcript and to get rid of the mutant enzyme, and we stop the production of the toxic product. So that's my Harrington project idea. Started 2 years ago. I still remember the first project meeting started 26 February 2022. And in the last 2 years, so we have screened 60 gapmer antisense and over 20 siRNA, and we found a list of candidates. Here is one of the example, and you show but we treat patients' fibroblasts, and we show very efficient and specific silencing of a mutant transcript, as shown here, which is in black, and we keep the wild type allele untouched, normal, which is in red. So this is in patients' fibroblasts, and we also confirm this is at RNA level. We also confirm at protein level. It's very promising. So we have the list of further candidates, and next step we need to move to in vivo study, because this is very new, especially allele-specific silencing. Working in vitro in cell doesn't mean it will work in vivo. We do need a suitable mouse model. But at that time, 2 years ago, there were 2 existing mouse models, but they both carry mouse gene, SPT-LC1 gene. As you know, it's a human-specific sequence, and mouse gene is it's useless. So we cannot use that 2 mouse model, and then immediately Harrington support me further to generate a humanized mouse. We do need, a model with a human sequence. At the same time, we also heard there is another heterozygous mouse model just generated in Oxford, and from Professor David Bennett's group. So we contacted Maryland, and very grateful. We got the mice immediately from Maryland Center, just in a month, free of charge. So we got the mice, we culture cell, and we design ASO in 3 months, and then we immediately we start the treatment in this mouse model. So we treated the neonatal group and we treat adult group. 7 days after, we collect liver and DRG. So you can see this, in liver and DRG, the mutant transcript significantly and specifically been downregulated, and then no effect on the wild-type transcript. It's very strong proof of concept, confirm this, the idea works, allele-specific silencing work in vivo. And then the next step, we repeat the treatment. Another experiment we did, we show is we need to do the weekly treatment. The treatment is subcutaneous injection. So we treat the mice weekly for 8 weeks, and at the end, we collect the liver, DRG, and sciatic nerves. So here we also include another group of antisense. So we have an unconjugated one. But think about how important the liver is involved in sphingolipid metabolism. So we include another group with gamma conjugation. It's completely new. We didn't know if it works or not. We just think to test how important liver is involved in this disease. So now you can see. And so in liver, we show over 90% silencing on the mutant transcript. And in DRG, it worked. In sciatic nerve, it also worked, although at a less extent. So and also another thing is very exciting is the gamma group, which is you see in green. It worked much better than the unconjugated group. It's very encouraging data at RNA level. And then how about functional level? Well, very lucky because DSP is a validated clinical biomarker. So we can use it directly and we test in our mice. So this is the data we got from our Swiss collaborator, Professor Thorsten Hornemann. You see the red one is a disease and the black one, lower one is wild type. Even from 7 days old mice, you already see the toxic product dramatically increased. So use it as a biomarker, and how about the treatment? After 4-week treatment in GANAC group, which is indicated by the red arrow, GANAC group significantly reduced, and further, there's 4 weeks treatment, after 8 weeks treatment, further reduced. It's very, encouraged by this data. And then at the same time, we got the humanized mouse model from Cygen, just in 6 months' time. Yeah, since we placed the order, by the time we got the mouse model, in 6 months' time. So this human, it's not complete genomic replacement. It's just the mouse exon 5 being replaced with human exon 5 carrying the heterozygous C133W mutation. So that one, we just can't wait to test our lead oligos. So we have some mice, and then this is a very pilot study did a few months ago. So we treat these mice. Once we have the mice, and we treat them weekly for 4 weeks. This is a GANAC ASO. See, in liver, so the level of the mutant transcript, over 90% reduction, and no effect on the wild-type allele in this liver and the DRG as well. 50% downregulation, that's 4 weeks. So this is some preliminary data we got. At the same time, actually we spend lots of time to characterize the new mouse model, because we need to also have some neuronal phenotype and to show the drug works. But we spend lots of time, and then at the end, we change, another idea is to change the diet. So after 6 months, the mice didn't show anything. But for drug screening, if you have to wait for too long, for academic lab, will make us bankrupt. And also the previous mouse model, we know you have to wait more than 12 months, and the mice start showing some phenotype, very mild phenotype. So what we did, we changed the diet to alanine. Remember, this SPT mutation is change the substrate from serine to alanine. So now we give the mice alanine to push the phenotype happen. So yeah, it works. This is and I got the data just last week, and this is a hot plate data. 3 months after alanine diet change, we saw the difference between wild type and the mutant, especially in female mice. So we sought out that the phenotype part, and this data I just received last night from Switzerland. I'm so excited. I just want to share with everyone. This is the DSB level in the humanized mouse model. The left-hand side, right-hand. I don't need to tell more. You will see the difference. Left-hand side, wild-type mice, and right-hand side, the humanized mouse model with the mutation. So yeah, it's kind of everything ready. We have the lead candidate. Now we have the humanized mouse model carrying the right mutations, the human sequence. We can test our oligos. So for next step, we will carry on with the detailed behavior study. So we will, using this precious mouse model, we will finish the in vivo study on efficacy, biodistribution, and toxicology study. So this is the next 2 years what we will focus on. And then hopefully in 2 years' time, that's in our timeline, we can move to clinical trial. I remember yesterday with Harrington talk about the 40 drugs. Hopefully we can contribute at least one of them. So this is the SPTL-C1 project. In summary, we complete the proof of concept using the surrogate mouse model, and also we generate a new mouse model, very suitable for test out the human ASO and siRNA as well. And also here, another important message we show here is the gamma conjugation. We didn't realize how important it is, but now we know. And also, as you know, gamma conjugated ASO, they have very good clinical translation, very promising clinical translation, much more than the unconjugated one, and the low dose and reduced toxicity. So that makes our project even more promising. And also the neonatal treatment or pre-symptomatic treatment. As you see, even 7 days mice, the toxic product already there. Should we still wait until the patient they have symptom, or we should treat them pre-symptomatic stage? So that's something we need a lot of thinking and discussion. Another thing we noticed here, the sciatic nerve target is still very challenging. And that's something we would like to carry on working with Oxford-Harrington Centre, and using the expertise on ASO delivery, so to sort out that, tackle that part. And I'm very grateful. For my excellent team. This project is led by Dr. Jin Hongmeng and another 2 research assistants. As you see, in 2 years, generate those data. You can imagine how much work it is. And also at the same time, I would like here, I take the stage and give my special thank you to my Harrington team for the excellent management, and Dr. Diana Westmore, and then also Michael Holland for the excellent project management. So this project is not just about the funding, it's also about project management and also the excellent advisory team advice I received from Harrington as a scholar. So that guided me in the last 2 years. I didn't waste my time. We didn't waste my just follow the drug development step by step and save lots of time, lots of energy, and to complete this project. Thank you very much for all the support. And then I still have 6 minutes. I'll talk about our work. So this is called UK Platform for Nucleic Acid Therapy, short for UPNAT, not UPNAT, it's the UPNAT. And this is, I think this morning, Cassie talked about the MRC rare disease platform. So this is a part of the platform. And last year, MRC and NIHR, they spent 40 million to support 11 rare disease nodes to promote rare disease research and to accelerate rare disease diagnosis and treatment. So we are one of the 11 nodes. So this is you can find all the detail about the 11 nodes. The 6 are disease specific, another 5 is cross-cutting. You can find all the detail from the website, RDR website. So for our UPNAT node, our aim is to bring all the stakeholders together and for knowledge exchange, data sharing, and to promote preclinical development and clinical translation of nucleic acid therapy. So our vision is to provide a roadmap for the rapid bench-to-bedside translation of this technology for rare disease treatment. So the node is physically based at UCL in key partnership with Oxford, Cambridge, NATA, and Genetic Alliance UK, Genomic England, and also another 5 NIHR BRCs. We have 3 major work packages. So work package 1 is focusing on target selection and preclinical development. So led by Professor Mina Wrighton, who is the director of Dementia Institute at Cambridge, and also Professor Alice Davidson from UCL. So that part, we have the working group specific on target selection and to generate the selection criteria. We focus on 4 parameters. So it's genetic variants, genes, disease, and patients. We don't think all this should be considered as a group, not isolatedly. At the same time, we also work with other international consortium and in collaboration with, here is N-of-1 collaborative. You have heard a lot from Professor Tim Yu. So they found this N-of-1 collaborative and they lead this consensus guideline, which will be available online next week. So the paper will. So here we collaborate with them and do our contribution. We assess this guideline and use the mutation we have to see how. Because the assessment we have done quite a few rounds of assessment. So we hope at the same time we also would like to generate some guidelines for the UK system as well to help this guideline can be implemented into our NHS highly specialized service. We already have a few to target and to test the new guidelines. And then second work package is to test UK's capability in nucleic acid therapy development and in key partnership with NATA. So we focus on model system of in vitro, in vivo validation using different model system and toxicology study and also manufacturing. So that one we collaborate with NATA and we already have 1 project, actually 2 projects, one on pediatric neurodevelopmental disorders with NATA and start from preclinical development, hopefully to trial. And also there's another project picked up by EOM for further clinical translation. At the same time, UPNAT also actively seeking partnership with other biotech, pharma, and the CRO to work on this, on this topic. The third part is led by Professor Francesco Montoni and Professor Carlo Rinaldi. So we focus on ethics, trial design, and the regulatory pathway. So that part we are in close partnership with Rare Therapy Launchpad and through RTLP's unique connection with government, with other organizations, with MHRA. We hope that part, and also we will do our contribution to the clinical translation and promote the clinical translation of nucleic acid therapy to a fast clinical trial. And also at the same time, we think about the training for junior doctor and next generation doctors and scientists. That's something that UPNAT has considered. So all this work we cannot do by ourselves and needs a great national, international collaboration effort. Today you already heard a lot from RTLP and N1C. So that's our UPNAT sitting as a door, why we call node. So we are the nodes connecting different parties, the international and international parties, to find the common ground and then found the synergies to work on in this area. And just like I'm very inspired by the talk today and yesterday. So just like Stefan, I don't know if Stefan's still here. I'm also a dreamer. So this is like Stefan, the map he showed, this is also our roadmap of UPNAT. Although I'm not good at singing, sorry, I forgot to include Royal Mail here. But this is a roadmap we would like to achieve in 5 years' time. Start from genetic diagnosis up to the drug treatment to patients. How long will this take? And the pioneer work from Professor Tim Yu already showed it's possible 1 year. For Milasen, about 1 year. Usually the traditional drug would take 20 years, 10 years. And Milasen take 1 year. This morning we have heard another task, 100 days challenge. So yeah, is it doable? I think together we can. We can, we will, and we must. Thank you. Thanks very much. Oh, yeah, some lovely picture we got a few months ago from the inaugural symposium of UPNAT at UCL. You can see lots of familiar faces here. And we also very welcome everyone to London next year, June, and also with the training, this time with a training workshop for junior doctors and scientists. Yeah, I finish here. Thanks all our partners. And